All right, so let's get back to some more mundane things. Sitabra showed you exciting stuff. Let's get back to trying to understand what happens to the dynamics in Hodgkin-Huxley, or what I will show you today, Morris-Lacar and so on. Let's ask ourselves, what are some of the behaviors that are actually possible in neurons, right? And it will turn out, I will show you, that there are more than one type of neurons, and the behaviors, even if you are considering only point neurons, even if you are considering only spherical cow neurons, can actually be quite interesting and diverse, right. I left you with this question, that what do you think the dynamical range of a Hodgkin-Huxley neuron is? So, if it can fire, what are some of the frequencies over which it can fire? Yeah, because if it if you ask yourself, is it able to respond to frequencies of stimuli that are very slow or very fast, the question is whether the Hodgkin-Huxley can keep pace with that frequency, right. For example, auditory waves, when we listen, will have a certain frequency. I wonder if anyone's worked out what the frequency is, or when you hear stuff, right. Think about it, and you should be able to, if there is a neuron that is responding to it with what is called a firing rate code, its frequency is keeping pace with the stimulus, then that neuron in principle at least should be able to fire that frequency. Would you agree with me? If your input frequency is coming at a higher frequency and the neuron is taking its time to fire, why do we think that this could be? This could be because of the refractory period. If the refractory period is so large that the neuron cannot fire fast enough, then you would miss information, right. It won't be able to respond to all of that stimulus. So, let us take a guess, pure coin tossing guess. Do you think a neuron can respond to arbitrary size frequencies, frequencies of arbitrary types, various frequencies, all of the useful stuff that we do in life, the neuron is able to respond to it? Well, you know the answer to this from the slowness side of things, right. If neurons are typically fast behaviors, right, ionic behaviors are typically fast behaviors on the order of a couple of milliseconds, right. So, what would correspond to something that changes on the order of minutes or tens of minutes? Lots and lots of processes in the body change on the order of tens of minutes. For example, hormones that are secreted. We just had breakfast, so our hormone insulin is stimulated at the moment, right, because there is glucose entering into the bloodstream, that is going to cause the pancreatic secretion of insulin. Insulin will eventually bring your glucose back down, right. So, right now we are all at a high from having had tea and biscuits, all of that sugar is circulating in the blood, right. But this process is going to take place on the order of several minutes, tens of minutes, hours, right. Do you think neuronal activity is useful directly in coordinating behavior on this time scale? By neuronal activity, I mean ion channel activity, electrical activity. Electrical activity is useful for mediating connections, movement of information on the order of a couple of milliseconds. Do you think a neuron is just going to stay active for tens of thousands of milliseconds? Is that how things would work? Well, clearly not. There will be other molecules that come into play. We saw some chemical processes like chemical diffusion, for example, right. Perhaps some diffusion processes come into play and so on, right. So, neuronal activity is going to have a role. It is going to have a role in perhaps coordinating, perhaps mediating, perhaps triggering the onset and offset of things. There is going to be a role, but there is going to be other processes that are going to be interacting with ionic channels. Does that make sense? Right. So, for this reason alone, we would like to ask, what is the dynamic range of a neuron? Now, does this question make a lot more sense to you, to ask why would we want to know what is the rate over which a neuron can respond, over which can fire. So, what is the firing rate capacity of a neuron? This is the question that I would like to answer over the next couple of minutes that we discuss this. Okay. So, I am also going to lay out a couple of open questions that are exercises for you to work on, right. Maybe you can make a little note for yourself that this is an exercise that I should work on. It would be a nice instructive thing to do. One of these is going to be, can we convert a Hodgkin-Huxley 4-dimensional equation to a 2-dimensional equation? I offer my help to those who want to do this, right. So, I am going to list out a few things. The Hodgkin-Huxley is a 4D model. Can you make it go down to 2D? Can you do something? Can you make certain approximations that you can actually represent this as a two-dimensional model? Okay. The other sort of models that we are going to that Chitabra pointed out were two-dimensional Fujinagumo models, right? So, these were 2D models, and Chitabra promised you that a Fujinagumo equation is a good representation of a Hodgkin-Huxley equation, right? So, how does this connect? Can you try and work out for yourself? How can you use understanding of the Fujinagumo in trying to understand what happens in the Hodgkin-Huxley? Okay, and the logical connect would be to pass from the Hodgkin-Huxley to a two-dimensional version of the Hodgkin-Huxley, and then ask what's different in the Fujinagumo with respect to the Hodgkin-Huxley. Okay, and the other kinds of question that we could ask is, instead of studying the Fujinagumo, I might study, for example, a new model which I am going to show you today, right. This model is going to be called the Morris-Lacquard model. It was a model that was developed by Morris and Lacquard, two neuroscientists who were working on the barnacle fiber. It's a giant barnacle, has a muscle fiber, and it was a lovely neuron to work with. See, most of these at the time, you're always limited by the experimental techniques that are available to you, right. So, if you find a fiber that is a nerve fiber that's actually quite big and nice to work on, then people start working on it, trying to understand the behavior of that and hope that whatever insight they get from that, from studying that, translates. So, Morris and Lacquard worked on the barnacle muscle fiber. Hodgkin and Huxley worked on the squid giant axon, which was a big muscle, it was a big neuron to work with, and they could do experiments with that and so on. So, these names are somewhat historical, but the pedagogical component of it should not distract you from the fact that we are learning something interesting and perhaps translatable to other systems. So, in fact, many of the neuron systems that we know today are going to be versions of these things, right. Perhaps there will be many ion channels that are involved. The cardiac cell, at last count, it had a dozen ion channels. How many channels does the cardiac cell have? The number of channels, different channel ionic currents that are there involved in the membrane potential of a cardiac cell. Yeah, right. So, I mean, if you want to know more, you can ask him about what are some of the behaviors of these things. So, there is a lot of neuronal models that have been developed, for which the ion channels have been characterized in great detail. There are also lots of systems for which they have not been characterized in great detail, right. It's both an evolutive and an involutive process to try and develop a gamut of a zoo of channels and membranes and what systems they apply to and so on, right. So, it's a whole exercise in itself. What we are trying to do is try to understand the basic features of neural models, and for which reason I will show you something you can do with the Morris-Lacquard. Okay. I am going to actually show you that there is two types of behavior that actually exist, which were originally identified by Hodgkin and Huxley themselves. They were called the class 1 neuron and the class 2 neuron. So, both of these class 1 and class 2 neuronal behaviors, which are different, show up in the Morris-Lacquard. So, the exercise that I want to carry out is to show you the Morris-Lacquard equations and then give you a facility to actually work with the Morris-Lacquard equations. If you wanted to work in the class 1 regime, then you can study that. If you want to study the class 2 regime, you can study that. Right. So, from the point of view of those of you who want to take on projects, this will be a tool that will help you get into it, right. So, I am going to give you all the parameters, I am going to give you all the files, and let us try and see what the features of these class 1 class 2 neurons are. Okay, that is one part of what we are doing over here. And then we talked a little bit about synapses, some of it in my lecture and some of it in Chitabra's lectures. So, synapses are what allow you to connect up networks of neurons. Okay, from synapses we start building networks, Networks, of course, is a very large field and it's very involved. What I'm going to do is, hopefully by the end of today, I'm going to give you a sense of what kind of behavior can start appearing in networks, and then I will leave it as an exercise for you. Perhaps some of you will do projects on this to explore what kinds of behaviors can arise in a network. Right? I'll show you some examples of what happens in a network. We'll walk through it in a hand waving sense. I'll give you a sense of what kinds of techniques and tools one can use to study networks, but then I'm going to lead you to study interesting problems, and we are here to help if anyone wants to get into that. Right? So that's my plan for what we are doing. This I will leave as an exercise. I might give you a suggestion how to do it. Right. So, if I am out of time, and just remind me very quickly, I will give you a suggestion of how you should go about doing that. Okay, shall we continue? Any questions? What the format is, where I am headed with this? Okay, as I said, the goal in this set of lectures is to give you actual tools, codes, files, techniques, working it out for you, so that you can do these things yourself. Okay, that's the main goal here. Okay, so let's talk about the famous equations of Kathleen Morris and Harold Lacar. It is a model with 3 channels. Okay, let me go back to this. It's a potassium leak channel. It has a potassium channel, a leak channel, and a calcium channel. In the simplest version of the models, these are the equations, right? The N stands for a potassium channel. You've got a calcium channel over here, the corresponding reversal potential for calcium. You got a leak channel, a potassium channel, right? The N is the gating variable corresponding to the potassium channel. We write a proper equation for it, a proper differential equation for it. Okay, don't worry so much about the phi. I'll use it, but it's a parameter that comes from temperature dependence, right? We won't worry so hard about it, but we'll use it in a way that we can manipulate stuff and so on. Okay, so let's those are the functions. We've seen things before that corresponds to the m. These two correspond to the n. That's the infinity function. That's the time constant. All of them depend on v. Those are the functions. They look forbidding, but actually they're not so bad. I won't get into the details of it, but it's not so bad. I mean, if you look at them, if you plot them out, they're actually very neat functions, very clean, very nice, smooth functions.
In the case of calcium on the loan, there is a V into V minus EC. Why
Correct.
Is that different from the other? All the other equations just have one V. Here it's quadratic in V.
Oh, this you mean this? No, this is saying L is a function of V.
Okay.
M infinity is a function of V. So that's not multiplying V.
Okay.
Any questions about this? So that's those things. I'll show you some parameters in a minute. But anyone want to ask me any questions about the structure of the equations? If you've now that we've seen the Hodgkin-Huxley, you should be relatively familiar, comfortable with it. It basically follows the same format that we've been developing all this while, right? This I app, of course, is a tonic driving current, as we've seen before. Yeah. Okay, and these are the ionic currents. Okay, any question? Okay, so I'm going to show you some parameters that are useful to study in this. Okay, let's see. Let's go straight to the point. I'm going to discuss these two classes. One is this set of parameters, and the second is this set of parameters. Okay, the parameters corresponding to this will be called type 1 parameters, and the parameters corresponding to this will be called type 2 parameters for historic reasons. And I'll show you what the reason why this is. It will become clearer a little bit later, right? So this stands for Hopf. We've seen a Hopf bifurcation before. This is a thing called the saddle node on a limit cycle. Right? We'll see what that means, but it just as a matter of terminology, I thought I'd introduce it, and I'll let you read about the homoclinic component of it on your own if you get interested in that. Okay. Any questions? All the parameters are listed. So, in principle at this point you should be thinking to yourself, or the easiest thing to do is write down an XPP file and put these parameters in and fire up XPP to see what the behavior could be. Yeah. Okay. So, let us do that. I am going to use the simulations that are described in Bard's book, and I am going to show you some. I will do these. I'll produce these files for you. I'll produce these pictures for you right now. But let's try and see what is going on over here. Okay. So these are the solutions of the Morris-Lacar equations. Parameters are listed in table 3.1, the Hopf case. So here's what Bard is saying. Bard says a small perturbation from rest decays to 0 from the resting state. So if this is your resting state, if you stimulate the neuron a little bit, how can you stimulate the neuron? There's a couple of ways. You can perturb the neuron by changing its initial conditions. That's quote unquote a stimulation as well. Or you can change the tonic current, right. So in this case, I am going to play around with the initial conditions that are driving the neuron. So the neuron is resting, a perturbation comes in and maybe raises the potential a little bit. Okay. What will happen to that potential? Can we guess? So your membrane potential has been lifted a little bit. Remember, think back to the threshold. This is the threshold. There's a threshold in this phenomenon, right? But let's ask ourselves, if you're lifted just a little bit, do you think you relax back, or do you think something else will happen? You will relax back. Why do we know this? Why are you so convinced that we should relax back?
Okay, so just a little, so it may not have crossed the threshold.
So if it may not have crossed the threshold. Okay. So here we are going back to the idea that if you cross, there's some sort of threshold. Is that a hard threshold or a soft threshold? It's a hard threshold. So there's a critical value. If you jump over it, you go into an action potential, a full blown action potential. So this is the heart of excitability. A small perturbation possibly relaxes back, which is why the system would be invariant to noise. They wouldn't change much. The resting potential would just, if there was a little bit of noise coming in, fluctuating things around, well, you would fluctuate a little bit, but every time this fluctuation would relax back. But if the fluctuation was large enough, beyond a certain critical point, you would actually get into a full blown action potential. So a bit of excitation is tolerated. I am deliberately using the word excitation in the sense that something is trying to push you up, but beyond a certain point, this goes into an action potential. Okay, so we are all good with this. If there is too much of excitation, well, you switch to an oscillatory mode. Does that make sense? That's a sort of canonical behavior that we've come to expect now from neurons. All okay? Okay, so let me ask you this then. Do you have any questions about this? I do. What about the amplitude? What about the amplitude of this thing? Can that change?
You showed yesterday.
Oh, I am not changing current. That's a good point. Thank you. So, I am not changing the current. I am not changing the tonic depolarization of the tonic stimulus. I am simply playing around with initial conditions. Maybe a perturbation came and jerked it into a different location. Maybe some.
Depolarization is usually constant for a specific neuron.
Okay. How many people would agree or disagree with this? How many people say that the level to which you depolarize is going to be the same, so you pretty much have the same amplitude of this neuron? All of the others disagree or just not committed? How many disagree with this? Oh, some people didn't raise hands, so they are just non-committal. They just don't know what it is right now. Okay, fine. So my question to you is, how can we study this, right? It's fine to make a speculation, it's fine to raise a hypothesis, but we need a facility to be able to study something like this. How should we study this? That's the question that I'm going to develop for you. Okay, so I'll first show you the simulation, and then I will actually show you how we can understand how we can answer this question. And that's my sense of what I mean by understanding it. Is this amplitude malleable or not? Could I have expected a different amplitude for the action potential? Okay, so let's do that. I'll switch over to this. Let's hope everything will work. since I am doing it in real time, ML2 dot ODE should work. Where is it? Where's my, here it is. Okay. Let's start this up and try out some of these things. So, that is a resting potential, right. We saw how to do this. There was an initial transient, I did not worry about that so much because I did initial condition last and I settled down into my steady state. Okay. Let us say I want to change some of the initial conditions. So, give me some thing that I should do. So, I want to change, I want to perturb this neuron. Let me, one perturbation could be I could just, the neuron is sitting at whatever this value is, I could raise it up a little bit. Let us try that. Okay. So, I am going to change, any suggestions how I should change this? I want to perturb it. So, how do I perturb it in XPP? Initial conditions. Okay, let us try initial conditions. Let us try some new initial conditions. So, voltage was minus 36 in the steady state. Let us make it say minus 20 maybe. At minus 20 I perturbed it too much, I believe, right. Perhaps I should perturb it less. Okay, so let me try some initial conditions. New again, instead of minus 20, let us make it minus 30. There it is. Okay. So, our resting level was somewhere over here. If I perturb it a little bit, I actually relax back. If I perturb it too much, and notice that I just perturbed the voltage equation, I did not even perturb the gating channel. Is it reasonable to do this? From the point of view of simulations, do you think I should have also perturbed the gating channel? Well, perhaps I could have, but it is much easier to think of the voltage being perturbed without changing the gating variable, because voltage will be driven by an IAP which is sort of fluctuating. So, there could be some line noise. So, I am actually simulating line noise in this case, right. Some input which is not perfectly stable, it is jittering around, and that alone could have jerked the voltage a little bit. Does that make sense? Can you combine this with the mathematics and the physical picture? You can sort of play with it together. So, in this case I am just saying the voltage was something and now the voltage got jerked up for some reason, and if I had to do it biophysically, I would say there was some sort of a line noise that drove the potential up a little bit. Make sense? Okay. So, now you see that there is some gap over here. There seems to be some threshold value over there, right? And we have to ask this question, well, to what extent is this threshold the only thing that matters in a neuron? If it is a firing rate code, see there are two major rate codes, coding scenarios that you can think of. You could have a firing rate code where information is encoded in the firing rate. Okay. So, you have a low stimulus, my firing rate is small. If I have higher stimulus, my firing rate is higher in proportion. So, if I have a relationship that says given a certain level of input, my firing rate correspondingly changes, that could be the basis of developing a firing rate code in the brain, right. The goal of all of this is to try and understand what is the firing rate properties corresponding to stimuli. Okay. So, the other possibility is you could say that this is not related to it is not a firing rate code. The information is encoded in particular spike timings, boom, 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 boom. So, it is the sequence of firing that is where information is encoded. So, these are called spike codes and firing rate codes. See? And it could be a temporal code or it could be a spatiotemporal code, and all of the lovely stuff that we study as neuroscientists or computational neuroscientists is to try and figure out what is the brain doing when it responds to stimulus and produces an action. Okay. If you at very high level we are looking at abstract behavior and abstract processes of thinking and so on. But at a very mechanical level, we also have to manage things like I want to touch this laptop. So, I should be able to go from here to here, move my hand up and move my hand down. I should be able to do that. Perhaps I can't do it sometimes. Give me an example why I might not be able to do this. Well, you're all too young, but my shoulder's frozen. I can't lift it up too much. Well, I have to find another way to do it. So, instead of I want to raise my hand to that height, I can't do it this way. So, maybe I can do it this way. I have to be able to alter my behavior as well, right. I have to be able to redirect behaviors that I could do previously, which I can't do now for some impairment reason. I have to be able to avoid doing what I could do and then find another way to do it. So, there is an adaptation to it as well, right. So, the brain is going to have to work out all of these things. Your cerebellum is involved, your hippocampus is involved, all of the stuff that will correspond to visual input will be involved, so the visual cortex will be involved. All of this has to work together. What's the code? How is it doing it, right? Okay. So, here's our look at changes in time of a particular variable. If you want, and I will leave this as an exercise to you, in your exercise section of your notebook, start writing down another of the exercises. What's the corresponding n doing? I haven't shown it to you here. I haven't shown you what the gating variable is doing, right? But you could easily do it. I can do it, but I'd like you to take a stab at it. See what the gating variable is doing. See if you can understand what the gating variable is doing. Okay. So, we have the tools, we have the facility to do it. I could open up another window and I could see what the gating variable is doing as a matter of this thing. So, this is the integration version of things. This is what you are watching, you are seeing what the solution of the differential equations is. Now, let us switch to the alternate version of looking at things, which is the phase plane method. Okay. So, that let me write that down in a little box over here. The other thing that we want to talk about is solutions of our systems or ODEs and how they correspond with phase plane dynamics. Okay. So, if you will recall, phase plane dynamics will give you null lines, they will give you diffraction fields, it will give you direction flows and so on. What will be missing is, what is missing? Time. So, you will miss time, but you get all sorts of other geometrical features that are actually interesting for us to understand. Okay, so, let us see if we can do a phase plane analysis of this. So, anyone want to suggest what I should do as I am about to do it? What should I do? I want to see the phase plane version of these things. What should I do in XPP? Well, I will open up another window and then maybe jump in whenever you feel like. That other window did not help me much, right? The other window is also V over T, V versus T. I have to change this window to do a phase plane. Okay, so what should I do? Here it is. Let us go to this thing called view axes and I will say 2D. On the x axis, let me put down V. On the y axis, let me put down N. And what would be good values for V? I have to set up my viewport so that I can see stuff. What would be good values of V? Quick quiz. What should I put the lower value of V to be? Minus 60, minus 70, minus 80, something like that, right? Okay, so let me just do minus, I don't know, 60. Let's go with that. And what's a good y min to have? What's the variable on the y axis? N, right? What should I take? What was n? The fraction of channels that are open. So, fraction goes between?
0 and 1.
Right. So, minus 0.1, just a little bit below 0. And what's an x max? So, you have to do this. You have to think about these things because otherwise you will not see the effective behavior, right? You have to think about what the values are, what's the effective range over which things are changing. Your variables are changing. Okay, for x max, let's do what, maybe 40, 50, 80, something like that. How high is this? Yeah, it goes to about 40. I'll just take 40. Let's just see. And then y max, it should be say around 1 or something. Let me start with 0.8 maybe. Okay. So, I get something that has this is V, this is N, it is roughly along the scale 0 to 1, N versus V. So, V is on the horizontal axis, V is on this axis over that range. I sort of picked out the range looking at what my solution was, and I have my viewport for the phase plane. Can we do something interesting with this? What should I do next for this? I want to look at a phase plane. What did Citabra teach you was the next thing to find out or look at? Null clines. Let us do a null clines. So, these are my null clines. How did I get that? Well, I went to null cline, where is it? Here null clines, okay, and I click new. So, that is just 2 keystrokes N N, and those are my null clines. Does it make sense? All good? So, you can see this. Now, what is this little thing that is over here? I think it is that trajectory. Okay. So, you can actually switch between these two. You can plot a trajectory over here, and you can see its phase plane version over there. Okay. All right. So, now let us examine trajectories in the phase plane and see what that looks like. So, before we do, since you have studied the Fujino-Gumo, can you see that this looks very much similar to the Fujino-Gumo? You have a nearly cubic null cline, and instead of having a straight line for this other null cline, you pretty much have something that goes nearly like a straight line. So, qualitatively things are different and quantitatively things will be different, but all of the tools that Citabra showed you for studying null clines and basically will translate over here, right. The pictures are almost the same. Can you make this transition in your head that the null clines of the Fujino-Gumo are nearly the same as the null clines that I am showing you for the Marshall-Karp-Hopf? Are you convinced of this? If you are not convinced, that is okay too. Let us do some more. Okay. So, before we try and see what kinds of trajectories will be there in this system, what would I mean when I say what kinds of trajectories are there in the system? I would like to be able to see how the V and the N change. That is a trajectory of the system, right. So, this is a trajectory of the system. I told you that I did not show you what the N is, but I could have if I wanted to, right. But this along with this N counterpart, if I plot what the V and the N are doing at different pieces of time, at different time snapshots, I will get a curve in that space, in the phase space, and that is my trajectory. All right. So, I could ask you directly, can you guess what the trajectories are? What would you say? You can say a few things about these trajectories even without going into the details, right. Well, okay, what can you say about this point over here?
That's the same.
That's a fixed point and it's stable, right? Yeah. Okay. So, one thing, so can we check that? Let's check it, right? Can you suggest what I should do to check it? I mean, I'm checking whether it's a stable fixed point. What should I do?
Set B and at some other point and check if they come to that point.
Yeah, that makes sense, right? So if I start, by start I mean if I provide initial conditions where, for example, I start at this position, I should come close to the, it will take infinite time, but you should come asymptotically close to the fixed point, right? And I should be able to do that in a neighborhood all around, right? So, let us do that. Can I do that in XPP? Yes, I can. So, let me go down to initial, let me go up to initial conditions and I will say, where is this? Mice. Okay. I switch back to this, I go, I will click. So, this seems, this looks good enough, right? Things around are actually going back in. This thing seems to look like a trajectory to me. This thing starts to look like a trajectory, like one of these guys, right? A little bit out and then it comes back in, right? So, a little bit out and then it comes back in. So, Okay, so we can do this. So I'm now starting to see that maybe I can build trajectories that are excitable by exploring space a little bit farther around from a neighborhood. So the idea of stability is to study a small neighborhood of where you are, right? And that's called local stability. So when you say a point is asymptotically stable or stable fixed point, you're studying in a neighborhood, in a small perturbation around, in a local neighborhood of that point, what's the behavior of trajectories, and they should compress towards the fixed point or relax back towards the fixed point, and that's what makes it a stable point. Okay, so at the moment how many fixed points do we have in this system? Just the one, right? How do we know this? Because they are shown by the crossings of the two null lines. Whenever you have a green and a orange null line cross, those will be the points of intersection. Those points of intersections are fixed point. We have to study the stability separately, but the crossings will tell you whether that it's a fixed point. There are certain systems in which you'll get two green null lines and they will overlap. Those are not, the null lines are the same variable, so those won't be fixed points. Or two orange components of the null lines, two orange pieces of the null line, that's not a fixed point. When a green and a blue, orange cross, that's a fixed point. Okay, so that's just a visual cue to remember how to look for fixed points. Okay, so let's get rid of this, and yeah, good, yeah.
So we assume that it's coming from.
Okay, so let's check that, right? Let's say, let me see if I can do this for you. Oh, that went up too far. Perhaps I should do this. Okay, let's take a look at this one. Okay, yeah. This trajectory, this last one that I plotted, I simply moved my other window and I plotted it, and that's what it looks like. I plotted means I just said, show me this curve. So I can, this one is this last one I plotted. So any trajectory that I want to plot, I can actually look at its time domain version of it, and that's what it is. Okay. Okay. So maybe this is not quite an action potential. Let's look a little bit harder. Okay, what's the next step that you want me to do as far as working with null lines is to go? What would be the next thing you would want to see? Yeah, you will get to limit cycles later if they are there. I just want to explore the phase space. What kind of behaviors are going to occur here?
The minimum, like the smallest point away, the point that's closest to the stable point, which causes a large excursion.
But there's no trajectories. I want to build some trajectories.
Yeah, so
click a lot of places? Okay, you can click in a lot of places. That's true, that's fair. Okay, so I'm going to show you something else. That's called a direction field. The direction field shows you, if you're at a given point, what's the direction and speed with which you're moving. So if you're actually over here, you're moving proportional to that arrow, and that arrow is almost horizontal but a little tilted. Okay, so you can see if you're moving from here, this is an arrow, not a very great arrow, but it's an arrow. Okay, so this is an arrow. That means trajectories are moving in that direction. That's the direction and velocity, direction and speed of that such a vector. Okay, it is proportional to how fast you're moving at that point. Longer is faster. That's correct. Okay, so if you look at the direction field, and the direction field was just two commands, right? D, F. If you look at this, can you see where you are likely to be moving faster and where you are likely to be moving slower, and can you make sense of that? So which are the regions which you would suggest are the trajectories are moving fast? Yeah, so things over here, for example, are moving fast. Things over here, for example, are moving fast, right? And now immediately your mind is running to the Fujinagumo, right? Okay, yeah, that's exactly what Sitabra said it should happen. But you can see that visually over here. What's happening over here? here, here, here. What are these dotty things? I mean, they're very small arrows, but why do you think things are so slow there? That's almost right. So as you're coming close to the fixed point, shouldn't your gone off?
Check. Lock on.
What happened? No, it's gone off. Ran out of battery. It's working. It's coming. I can't tell. Is it at the back? Can you hear me? Okay. Okay, so as you come close to a fixed point, things are nice and slow, right? When you're very close to a fixed point, you expect to be slow, right? Because you're never going to cross. I mean, as you come closer and closer to when you cross an alkaline, you cross it only in one direction, right? As you come close to something where neither the directions are positive, you're going to slow down, right? So it's expected that you're going to slow down over here as you're close to fixed point, and as you're far away. But there's another interesting thing that's happening over here, right? You're actually going relatively fast when you're in this space and relatively fast in that space, but there's also something to notice, that you're moving in this direction and you're moving in that direction when you're going down. So that sort of says that there's something that you might be able to get as something cycling going on, right? And you know this from the Fitzgerald. You worked it out with sines and all of those stuff. A similar thing is happening over here. Okay, so now let's ask, what are some of the trajectories? And as was helpfully suggested, let's pick lots of points and do it. Okay. Okay. Let's do that. Okay, so that's another pair of commands, and those are some of the trajectories that you can see in this system. Okay, you can see things are going to spiral in over here, and there's a general spiral over here, but there are all these trajectories sort of go very far out before they recover here, right? So if you were to make a perturbation here, what do you think will happen? You'll get a spike, right? All of this. So you're here. If you cross over to this side into this region, you will take a big loop and come back. If you move a little bit here, well, you just sort of come back right away. This also shows there are multiple amplitudes. Yes, there's a whole range of amplitudes over here that look spiky-ish. Does it make sense? There are also amplitudes of that size. This is the amplitude direction. You can see something with a smaller amplitude, larger, any very large amplitude. So amplitude in this model is a malleable thing. Okay, I'll leave it as an exercise. Again, add a little note for yourself. Show trajectories of various amplitudes in this. Okay, does that make sense? Okay, so we've got now a facility for asking ourselves, how can we study various trajectories of this system? If you look into the phase plane, you get a range of trajectories all in one go, which you did not get from looking at the time domain simulation. So these two pictures are always useful to provide different kinds of information that are complementary in trying to understand your system. Yes.
So there are no explicit constraints which is fixing the n from 0 to 1. It can go to minus 0.
Yes, that's correct. So his question is, for those who didn't hear it, what about negative n? Should I have bothered about negative n? And the correct answer is, he's right in saying n should not go below 0. So technically, anything below this level is an unphysiological solution. It's part of the mathematical solution, but it's unphysiological from the point of view of the physics of it, the biophysics of it. So our standard tactic for handling that is, well, let there be a mathematical solution, but we're going to chop it all off and say that I am only interested in things that are above 0 as far as the n is concerned. And so also for things that that might, for example, lie outside of 1. It's the physical constraint on it that sets out for us what the behavior is going to be. in what region we should look. Okay. The next thing that I want to study, any questions on this? The next thing that we wanted to study was this. So, now given that I show you the phase plane, can you suggest what I should do in order to get continuous spiking? None of the sorry, just one moment, none of these trajectories are spiking continuously, right. They all spike at different amplitudes with different so to speak delays and so on, and they all come lengths and so on, and they all come back and end up over here, right. So, no matter where you are in this phase space, you end up there. So, this fixed point is not only locally stable, it is actually globally stable in the region of phase space that we are interested in. Okay. How can we get spiking? Now, introduce current. Okay. So, great. So, what should I do to the current? Should I increase it or decrease it? Okay. So, let us go back to the null line picture and ask why should we, from a physical point of view, from a biophysical point of view, it makes sense to excite something in order to get more activity, right. Can you see that from a mathematical perspective? From a phase plane perspective, from math, what do you think is going to happen if I inject current? Which null line would shift?
Oh, sorry. Orange is the voltage only.
The orange is the voltage null line. What would happen in the voltage null line if I add current to it, positive current to it? It should shift up, right? Can you convince yourself of that? Yeah. Okay. Let us try it out. So, I am going to change the parameter. I am going to make it I app. It is at 60. Let us make it say 90, and let us plot this null line. What will happen to the red to the green null line? Nothing will happen, right? Why? Because the I is added to which equation? It is added to the voltage equation. So, if it is only there in the voltage equation right hand side, right. So, the only curve that can change is that curve, because it is not there in the right hand side of the n equation, it will not change anything. Okay. Let us hope that happens. So, this is what you see. Okay. So, you were here when you added some more current to it, you actually lifted the orange null line. This tactic of adding current and seeing what happens to the or raising the voltage null line when you add a positive current and suppressing it, I can actually make it 30 and lower the null line also, right. You will all agree. Okay. So, let me just do that. Now, I have reduced the null line, and there it is. So, I have reduced the current, and so actually I get a null line which is over here. I can set it up so that I raise this null line like so. So, this intersection is on the middle branch of the cubic. We could do that, right. Another exercise for you, try doing that and see if you can get an oscillation going. Okay. So, this point that is here, you want to study its stability also. So, there is another way to do that. Okay. There is a way in XPP to do this. Let me see if I can show you that. That is my fixed point, right, at the intersection. Okay. So, let me go and show you that you can click on this thing called singular point and click on this thing called mouse, and it is very helpfully it says click on where you think the fixed point is. So, I am going to go over and I am going to click on this. I think my fixed point is somewhere over here. So, I will click on it. It says do you want to print the eigenvalues? Okay. Let us print them. Okay. We will discuss this a little bit later if anyone is interested, but at the moment I am not interested. I just want to look at this thing that says stable over here. So, it is going to be a stable fixed point. Okay. It finds that fixed point for you, and if you look on the window over here, it will tell you exactly what the values are. Tell me what the eigenvalues are. Huh? Yes, it is a numerical eigenvalue estimation, that is right.
It's not using some symbolic.
No, this is all numerics here. Yeah. So, it finds you the fixed point that you want to know, and it tells you that it is stable. Okay. So, you can play around with another exercise, add more current. At what point will the with this become unstable? We can do this in a couple of ways. We can actually do change a lot of i's and find out where the stability is, or you can switch to the bifurcation diagram. Okay. So, shall we go over to the bifurcation diagram now? Okay. So, let us compute the bifurcation diagram for this thing. How do I do that? I app, let me start at say maybe 50 or something. I think it is a fixed point at 50, right? Yeah. Okay, so I have added a current I app equal to 50 and I get a voltage that is here. I did a couple of initial condition last to get a nice stable fixed point, right, to be really close to the fixed point. Now I can switch to auto. So, let us do this. I have opened up auto, and remember that there is a certain sequence and we just walk down these buttons in auto. Perhaps I should have closed all the other windows, or maybe I will just leave them in the background. So, let us see if I can do this. The parameter I am going to use is IAP. I am trying to produce a bifurcation diagram to see all of the behaviors that are present in this model. Okay. We have seen the behavior of a Hodgkin-Huxley neuron, right? Can you use that to predict what might happen here? Okay. Recall the bifurcation diagram of the Hodgkin-Huxley that I showed you last time. Let us ask whether we can get this over here also, or whether I expect a similar thing to happen over here. Let me see what is my maximum that I want to go to. Should I go to 200? Maybe. I don't know. Maybe I should go a little bit more. Are you able to follow what I am doing here? Okay, right. If anyone is not quite following what we did, again I will have a tutorial session in the evening today. You can come back and check with me, right? Try it yourself and then I can show it to you. So, this is from here to here. What do you think this is? A branch of stable branch. This is unstable branch and this is stable branch. Okay. And let us move along this grab points, move along. This point 3 over here is identified as a Hopf bifurcation. Yeah, okay, that is nice. Let us move along further. Do you think this point 10 is a Hopf bifurcation also? Let us check it. Okay, it is. This point over here is also a Hopf bifurcation. So, around a Hopf bifurcation I expect what? Periodics, right? Yeah. So, I expect periodics. Let us try and bring out the branch of periodics in this model. Okay. I am going to go to numerics, make it ds minus. Okay, I want to move backwards. I mean lowering IAP. So, let me now run periodic. Okay. There it is. So, sometimes sometimes when you run auto, I will warn you, sometimes it will find a branch and then go back and do the same branch again, you will just keep doing that a little bit, right. Like it might go and find this branch, then it comes back and finds that branch again, then goes back and does this branch, and when you run into a problem like that, this little abort button is what you do. Okay. Okay, so now let us take a look at this. What do you think this branch is and this branch is? The blue stuff. Those are branches of, those are periodic orbits, but are they stable or unstable? They are unstable periodic orbits. And this one, this green branch is actually a branch of stable periodic orbits. Okay. So, shall we take a look at one of these trajectories and see what it looks like? Okay, I am going to go grab something along the green branch if I can find it. Oh, there it is. Okay, let us take a look at this, right. So, it is this oscillation that I'd like to pick up. So, I'm going to grab it and I'm going to watch it in the time domain to see what it looks like. Okay. So, that is a nice big oscillation. Let us continue for say 400. That is an oscillation. Okay. So, that particular oscillation that I had picked in auto is actually looks like this in the time domain. Can you convince yourself that it has the right amplitude, so that I have the right trajectory? It goes from say 35 to minus 52, that's minus 50 something, 32. I think it's right. Okay. And is this trajectory going to be stable or unstable? It's going to be stable, right? It comes from that green branch of periodic, so it's going to be stable. Okay, all good? Okay, so this looks similar to what we had for the Hodgkin-Huxley, except that you have this this lovely blue-green thing happening over there. So in the Hodgkin-Huxley, it was a supercritical hub, and you got the green branch alone, but you didn't get this blue branch. But it looks similar to us, right? You got a bubble of oscillations around an unstable branch with two strings of it's like a sausage with two strings of stable branches. Okay, all good? Okay, now let us get to the main question that we had promised we would answer. What is the dynamic range of this neuron? What are the frequencies over which this neuron can respond? How do we get that? Well, can you get the frequency from this curve?
Find all the peaks.
Find all the peaks, that is time period, 1 over time period will give you the frequency. Okay. So, you can do this for a whole bunch of, where would you change your current from to from, where would you go from to find all the various frequencies? Do you think the frequency here is different from the frequency here? I do not know, we will have to check, right. Can you tell it from this graph? No, right. From that graph you will know the amplitude. I still don't have frequency information though. So, I would have to pick different positions, pick out various trajectories and plot the frequencies at position 1 and position, when I say position I mean current 1, current 2, current 3, and the effective range over which I have to check, this is the only range over which you can get oscillations. Should I check here? Well, there is no point, right. Should I check over here? No point. I know immediately from the bifurcation diagram there is no oscillation at that level, there is no stable oscillation possible at that current, right. These if you pick your currents very, very carefully to be in this region, you can actually get unstable orbits as well, unstable periodic orbits. Okay. So, if you are very careful, another exercise for you, zoom in on this region and see where you can establish there is actually bistability in the system at this point for that region. Why would that be? What would be the two stable attractors there, two stable behaviors that you can have?
One of the red points.
Yeah, one of the red points here and a corresponding green point over here. So, again zoom in on it, play around with XPP, push your current along the curve to be exactly there in this domain, and then you can show that there is bistability in the system. So, this is the region of bistability. You can see that from here to here in this region you can have a stable fixed point, or you can have a stable periodic orbit, a limit cycle also. So, yeah, play around with that, another fun exercise to do to see if you can find what the trajectories are.
So basically some sort of like resistance, you go this way and it will give you limit cycle, but if you take down the I applied, it will still be in a limit cycle and it will come down later.
Yeah, that's correct. So, what he's suggesting is perhaps you can use this setup to perform a sort of hysteresis in this, and the answer is yes. You will have to drive your current into the appropriate region. Let us say you drive your current into this region, it will give you if you drive it up here, you will be in limit, you will be oscillating. If I drive it back down, you will still be oscillating slowly if I drive it back down, if I drive my current back down, it will be oscillating, oscillating, oscillating. If I drive it up here, boom, you are in a stable position. In your stable position, keep going, keep going, keep going, you'll stay along the stable direction, keep turning up the current here, boom, now you are in an oscillation. And you can set up some cycle which corresponds to hysteresis. Okay, for those of you who know what hysteresis is. You can do that. Lots of models actually rely on this. Lots of calcium based models of say bursting often rely on this. So, that's another thing to study. Perhaps you can do it for one of your projects. You can study what bursting is. Right? Bursting is not just a single spike, but a series of spikes, grrr, and then stop, grrr, then stop. So, you can study bursting by using hysteresis ideas.
So, when a neuron is on the top, like the tetanic stimulation region,
Hmm.
and we inhibit it, we can push it into an oscillatory phenomenon, right? Where
Right. So, in this
the top
model,
side-
oh, sorry, what did I do? Oh, I think I lost it. Give me 1 minute. Yeah. Sorry. Right. So, if I go into a if I run into a current that's high enough, I saturate the neuron. If I turn down the current, I'll restore oscillations. Okay, any questions on this? Okay, yeah, yeah,
The blue line, it was like
yes,
why was that pattern?
yes. This is something that people who study dynamics recognize. So there's two things that can happen. You assume passage from the stationary branch, so stable branch to the unstable branch. The mouth of the crocodile could be that way, or the mouth of the crocodile could be that way. When the mouth of the crocodile is this way, it's called a subcritical Hopf bifurcation. If the mouth of the crocodile is that way, which you saw in the Hodgkin-Huxley model, it's called a supercritical bifurcation. So you're right in noticing that this is a subcritical Hopf bifurcation, but what comes out of it is a branch of unstable periodic orbits, and they turn around into a branch of periodic orbits in a joint, in a limit point. Why does it happen? Okay, so the answer is going to be a little bit more involved depending on whether you expect a mathematical answer or a biological answer. From the point of view of mathematical answers, there's only a few things that can happen. So this is one of the things that can happen. From the point of view of biology, it's a little bit harder to describe what would a unstable periodic orbit mean in that system. If things are very, very finely tuned, you can actually go around in a circle and come back to where it is, right? So I'll leave that as an exercise for you to work out. Okay, any other questions on this? Okay, so let's get back to our holy grail. The burning question before us is, what's the range over which this neuron can respond? Okay, so let me go back to this and show you the frequency curve. You saw the range of oscillations. Let's do frequency. There it is. Okay, ignore these unstable branches, right? What can you look at so this is the frequency as a function of phi. So this is the bifurcation diagram, except you're describing frequency. Okay. What immediately strikes you in this diagram? That the range of frequencies, no matter how you change the current, is actually quite limited, right? In particular, it cannot fire at very low frequencies, and it cannot fire at very high frequencies, right? This neuron is actually band limited in the frequency to which it can respond, with which it can respond. Okay. So is this a good neuron for you for studying firing rate codes? Well, the answer could be depending on what you do with it, right? If if you are in this region where your frequency is tuned to what the stimulus frequency is, then perhaps you can make it resonate with that and do good stuff with it.
We can have multiple such neurons, and we can have a mechanical way of transforming frequencies to each other, and we can have neurons responding to a fixed.
So that's a great project idea, right? So you can try and see what would happen if you connect two neurons with synapses. What would be the frequency response of the coupled neuron system, right. Let us say a neuron is driving through inhibition a second neuron, or if that neuron were to drive that second neuron, but through an excitation. You have all of the equations for it. What would it would the coupled system have, or would the second neuron have a larger frequency range, or would it have a lower frequency range? I don't know the answer. Maybe you can find out. Okay, so this is how you study all of the features of neurons that you would like to study. Okay, all right. So shall we go on? Shall we study some more? Do you want to take a 5-minute break, or shall we just keep going? Maybe a 5-minute. We'll stay here just a couple of minutes. I'll set up my machine for the next set of simulations.